Welcome friends to our channel Homeopathic Lighters. Today we will discuss the topic inflammation, both acute and chronic inflammation. We will be studying accordingly. So what is the definition of inflammation? Inflammation is the local response of living tissue to any injurious agent. It is protective response needed for survival. That means it is the protective response of our body that is against a injurious agent.
These types of inflammation, that is acute inflammation and chronic inflammation, we discuss by differentiating both acute and chronic inflammation. First is the onset. So acute inflammation, it is rapid in minutes per hour, whereas in chronic it is a slow days. Then cells, what are the cell informed in the acute? That is neutrophil. In the chronic case, that is lymphocyte, monocyte and macrophage. Then what is the tissue injury in the acute? That is mild, self-limited, whereas in chronic that is severe and progressive. And what is the local symptom which appear in the acute? That is prominent, whereas in the chronic it is less prominent. Then the 5 Rs.
The 5 R's that occur in inflammation are: first is recognition, that is the injurious agent's recognition. Then recruitment of the inflammatory cells. Third is removal of injurious agent. Fourth is regulation, means control of the inflammatory response. 5 is repair.
Then we discuss what is the changes occur in the acute inflammation. First, then why vascular changes, the blood flow increases because to bring the cells and the protein to the site of injury. And then what happened? By the vasodilation and increased vascular permeability, the cells and the proteins enter to the site of injury. Whereas in the cellular event, what happened in the acute inflammation? There will be requirement of a leukocyte occur. Why there will be requirement? Because of activation of a leukocyte leading to the process of destruction of the invaders and the production of the mediator by phagocytosis method.
Hence, vascular changes occur for the requirement of the proteins for at the site of injury, whereas the cellular events occur for the destruction of microagents.
So that is what are the stimuli required for acute inflammation. If the stimuli may be infectious, may be tissue necrosis, may be trauma or physical or chemical agents, may be foreign bodies, may be immune reaction. Then
how the stimuli recognized by host? The stimuli are recognized by receptors present on the antigenic presenting cell.
So this receptor, what type of a receptor we found? Main is a toll-like receptor and a inflammasome. That is a toll-like receptor is what? It is a microbial sensor present in the plasma membrane and endosome. What is plasma membrane? Plasma membrane, we know the cell outermost layer is plasma membrane, and endosome means the nucleus. In the nucleus, the content which is known as endosome. In the inflammasome, what?
Multiprotein cytoplasmic complex. Inflammasomes are multiprotein cytoplasmic complex. It recognizes the products of the dead cells. Okay. In toll-like receptor, recognize products of bacteria, virus and other pathogens. Means almost all types of pathogen. Then it results in production of mediators of inflammation, whereas inflammasomes activates the caspase 1, which in control activate the interleukin 1, which is the mediator of the leukocyte requirement. These leukocyte phagocyte and destroy the dead cells.
And now this is the stimuli, how it is recognized by the receptor. Okay, then we go towards the vascular changes of acute inflammation. So first changes is the changes in the blood flow and caliber.
As soon as the tissue exposed to injurious agents, the stimuli to the vasodilation of artery occurs, which is by histamine. Occur which causes the blood flow increases, causes redness. Okay, the first causes redness. Then it increases the vascular permeability, for which escape of fluid to the extracellular space occur. Okay, then due to vasodilation and increased vascular permeability, what happens? This combined effect of the tissue to causes slow blood flow and concentration of RBC, which results in increased viscosity. Then engagement of small vessels occur. Then stasis. Stasis means it seems in vascular congestion and redness of the tissue.
Means there is stasis, means blood flow becomes static, means low. So due to the increase in vascular permeability, the fluid which are present, they are passes from the blood to the interstitial tissue. So what is the mechanism occur in the increased vascular permeability? First mechanism is the contraction of the endothelial cell. So how you can see this increased contractility? What happened? By the contraction of a gap between endothelial cell increases. So here you can see. So first what happened? The contraction will occur. Then what if there will be gap will be increases through which the fluid is escaped. It is the most common mechanism, and it occurs immediately and is short-lived. Why? Because the histamine, bradykinin are all important chemical mediator which causes this mechanism in a shortly. It is also referred as immediate transient response because it occurs in immediately and a short-lived. That's why it is a immediate and a transient response. Then
the second is direct damage to the endothelium. Means in burns, exposure to the microbes, toxin, they directly damage the endothelial cells. Here you can see that the endothelial cells become divided to small particles. So what happens? It occurs immediately and is sustained for several hours until the damage is repaired. The fluid escapes through these damaged endothelial cells. Here you can see this.
Then endothelial injury due to the leukocyte. So what happened? In that time the leukocyte, that is mainly neutrophil, adhering to the endothelium may injure it in this case, and they causes the damage of the endothelial cell, so that it occurs immediately and it is sustained for several hours until the damage is repaired. Then transcytosis, where what happened? Transcytosis means increase in a transport of the fluid through intracellular channel. These channels are stimulated by two things, that is VEGF, that is vascular endothelial growth factor, which promotes the leakage.
We just finished our vascular events of acute inflammation. Now we will discuss the cellular events in acute inflammation. So here we will discuss about the recruitment of leukocytes. In the lumen, what happens? Like margination, rolling and adhesion. Then across the vessel wall, what happens? Then outside the vessel wall, what happens? Like chemotaxis. Last is what is the therapeutic of cellular event.
So first is the component of the cellular event. What are the influx of the leukocyte, that is mainly neutrophil and monocyte to the site of injury occur. First thing, what happened in the cellular event, that neutrophil and monocyte are enter to the site of the injury. These leukocytes ingest and destroy the injurious agent. Which injurious agent? Those microbes, necrotized tissues, foreign bodies, these are the ingested by these leukocytes. Then these leukocytes secretes the growth factor for the needed of the repairing. Those substance which are injured by these leukocytes, they also repair this by secreting the growth factor. Then how does the leukocyte travel from the vessel lumen to the injury?
It is a multi-step process and mediated and controlled by adhesion molecules and chemokines. So two things, adhesion molecules and chemokines. Here the chemokines are chemoattractants, that is cytokines, which play a role in leukocyte migration.
We discussed in the lumen how the cell, how the blood, that cell, that is neutrophil and leukocyte, enter to the interstitial too. So this process, that is margination, rolling and adhesion, now we discuss. So in the journey it is divided into three stages. What are things? First thing is in the lumen of the blood vessel, then migration across the endothelium, then migration in the tissue towards the stimulus. So if we discuss by taking the example, this is the blood vessel, this is the lumen, this is the capillary of the blood vessel, then this is the endothelial lining, this is endothelium line, then this is the injurious cell. Means this blood vessel, from this blood vessel, those monocyte neutrophil which are coming, they first from this blood vessel they enter towards the outside, then it will go towards the stimulus area. So how this occur? First we discuss, that is in the lumen what happened. Margination means where the blood flow sluggish or slow down.
Previously, we discussed that due to vasodilation and increased vascular permeability, the blood flow slows down. So then margination occur. Here, the blood flow slows down. So the central leukocyte moves to the periphery. That means the leukocyte which present in the blood comes to the periphery of the blood vessels. So gather along endothelial cell. It is basically the redistribution of leukocyte along the margin of blood vessel.
Then rolling. In the rolling case, what happened means first we discuss the margination, how they entangle with our endothelial cell. Now from this endothelial cell they get attached by leukocyte transiently bind to the endothelial cell and detach and again bind. This process is known as rolling. This process is mediated by a set of a protein. This protein is known as a selectin protein. Here you can see the blood vessel. These are the monocyte or neutrophil. First they what happened, they casually blood flow will be occur, but when there will be infection they get migrated to the endothelial lining. Then what happened, there will be rolling will be occur. Here you can see this is the rolling occur, and after the rolling there will be adhesion occur. Third step is the adhesion. Fourth step is the transmigration, means this particle now enter towards the extracellular fluid through the transmigration, which is known as a diapedesis. So in the rolling what happened, here you can see this is the WBC. Which type of WBC? That is neutrophil. Neutrophil having a receptor, that is a Cellular Lewis X, which get added to the selectin material of the endothelium, which is the interaction is a loose interaction. So that what happened, rolling what happened, rolling in the case where the decrease in speed occur and which results to the rolling. There will be selectin, which is mainly three types: P, E, L. What is PEL? P means expressed in the platelet, then E means expressed in the endothelial cell, and L means expressed in the lymphocyte or leukocyte. So these are the types of the selectin which present on the endothelial cell.
The P and E found in the activated endothelial cells, like it is by mediators like interleukin 1, necrotic factor, etc. Whereas L found in the surface of leukocyte. What the causes? The corresponding ligands are sialylated oligosaccharides, example Cellular Lewis X, that is low affinity interaction. Here low affinity interaction occurs.
Now we discuss that
now leukocytes know something goes wrong with body. As the leukocyte goes to this stage, they know that something goes wrong with body, so they express a protein integrin. At the same time, the endothelial cells which are activated express ligands, example integrin ligand.
Monocyte neutrophil, it is having the integrin, and here the endothelial cell which having the adhesion molecule, which is ICAM, that is intracellular adhesion molecule. So this type of integrin is called Cd2 Integrin, which appear. So integrin means what? Once the integrin bind to the integrinal ligand, the leukocyte adhere to the endothelium very firmly. Whereas in the rolling case, what happened? We see the loose adhesion, but in the integrin adhesion is a firm adhesion. What is integrin? These are the basically transmembrane glycoproteins involved in the adhesion. These are expressed only when leukocytes are activated by the presence of chemokinins. So there are, we discuss what type of integrin we discuss, that is ICAM and VCAM, which are present in the where? It is present in the endothelium under the influence of the chemical factor, that is human necrotic factor and IL-1. Then immediately after the adhesion, the leukocyte tends to squeeze in the endothelial cell. This process squeezing is known as a diapedesis and a transmigration. So that is our fourth step, where you can see the there will be this integrin when adhesion occur, then it comes out from this cell blood vessel by diapedesis. Okay. So this brought by proteins in the leukocyte endothelial cell, which is known as a PECAM1, that is a member of IgG protein CD31.
Pass through endothelial cells, they tends to secrete collagenase that breaks the basement membrane and enters to extravascular space.
This way we discuss all about when they break the basement membrane, then enter to the extravascular space. So what is the journey in the extravascular pathway, we discuss in the next video. Thank you.